Mhm. Great. Okay, so let's start with the introduction. Mm. Sora, please, can you read?
Okay. Which cells organs is involved in intracellular digestion and waste disposal? A. Liposomes. B. Golgi apparatus. C. Nucleus. D. Lysosomes.
Thank you very much, Sora. There's a few more seconds. Let's do poll. One more person. Okay, perfect. All of you went for B, what is absolutely correct. Uh-huh. Guys. Okay. There was some echo maybe, but everyone is muted. I don't know what's wrong. Hmm, I don't know. Okay, lysosomes. You went for correct option. Great. Um, absolutely correct. So, Sora, please, can you maybe remind us the lysosomes as an organelle?
Lysosomes is like garbage in the cells.
Mhm.
Then yeah, that's all.
Yes, that's absolutely correct. And I just want to provide you with the more understanding uh, about those analogies we used for Golgi apparatus, also for endoplasmic reticulum, also for lysosome. Guys, it's absolutely okay, and also for me, I really appreciate it that it's helpful for you to understand and to remember the organelles as their analogy from macro real world experience, such as you mentioned just now, the lysosome, something like trash can. Nevertheless, in the upcoming or in the future you will deal with another teachers and other professors, and they maybe would not be very satisfied if they hear something like this. For me it's absolutely okay because I told you that to how to remember it. But for the future it's just an advice for a future that try to maybe describe it even more. What do you know about that? What does it mean that lysosomes are trash can, for example, or garbage bag, or garbage tank? What does it mean? So basically, they are responsible for what? For intracellular digestion, waste disposal, or you can say also recycling material inside of a cell, and or degradation or unnecessary materials, products, substances whatsoever. Okay, so what I want to emphasize or highlight is that maybe you don't have to do it now, but for the future try to be more exact, or use medical terminology for the future. Okay, uh, we are just training for that. We are just practicing it now. So here it's absolutely okay, and I'm really glad that you are using those analogies. But we can also practice another, a little bit already higher step, how to express our thoughts or knowledge. Okay, so very good, but very good. Lysosomes were correct, and you you explained it as we learned it. So that's thumbs up. Um, yes, nucleus. We will get there tomorrow or day after tomorrow. We still haven't covered it so in depth. So nucleus, however, is not responsible for the intracellular digestion or waste disposal. What about Golgi apparatus, guys?
Modified protein.
Perfect, great. Proteins, or let's say uh, um, other substances, also lipids, for example. Yeah, also lipids are sent or produced within the cell, and also carbohydrates can be produced within the cell, and then it's the Golgi apparatus, as we said, is like a post office, and it delivers and it's sending, modifying those products. Okay, so egg perfect. Proteins is absolutely correct, but additional information, also lipids or carbohydrates, can be modified even in Golgi apparatus. Mhm. And then it's sorting, packaging, or also sending. The Golgi apparatus is also responsible for that. Great. Very well. So far, so good, guys. Very, very well. And the last one is A, ribosomes. What do we know about ribosomes? Ribosomes.
Ribosome synthesize protein.
Perfect. Ribosomes are responsible for protein synthesis. So they occur also in as well as in eukaryotes, as well as in prokaryotes. And there are some differences between prokaryotic ribosomes and eukaryotic ribosomes. Let me show you the picture. But what we have emphasized, what we highlighted, was basically they differ in the size. Prokaryotic are a little bit smaller. We mark them as 70s, eukaryotic 80s, but those numbers you don't have to remember definitely, okay? So don't bother with those numbers. The difference is basically they have some structural differences uh, in prokaryotes and eukaryotes, and prokaryotic ribosomes are basically smaller. That's all what we have to know. So basically, there's two things. Yes, Yuki, are you raising hand? Yuki?
Sorry, could you say it
Oh,
again?
I thought that you are raising hand. I thought that you want to ask a question that you were doing this,
Yes.
but you don't have question.
Oh, okay.
Don't have you don't have question?
Ah, yes.
Okay, so I guess no then. Okay. Uh, okay. So let's continue. Uh, that was the first question. Mhm. So also ribosomes are not correct for this question. Okay, let's do another one. Shoko, please, can you read?
Alright, how many layers of phospholipids does a eukaryotic cell contain? A 4, B 3, C 2, D 1.
Mhm. Perfect. Thank you. Two more people. Okay. Okay, so here are the results. Let's discuss them. Shoko, what have you chosen and why?
I choose C
Mm-hmm.
because eukaryotic cell membrane has two layers.
Mm-hmm. Yes,
Yes.
exactly. So let me just remind you the picture we saw during last week. Here we are able to see phospholipid bilayer of cell membrane, and on the right side of the slide we have it in more detailed perspective. We have a phospholipid heads and phospholipid tails inside. As you see, they are orientated in the opposite directions. So extracellularly, we have heads, however, also intracellularly, or on the cytoplasm side, we have the heads on the border or limitation to the space or environment. Do you know, as we see, we have two layers. Do you know uh, if the heads are polar or non-polar regarding chemistry?
I think
Hmm.
the head is polar.
Yes, the heads are polar and the tails are non-polar. So that's the reason why polar molecules cannot pass through the phospholipid bilayer, because here we have a non-polar part of the phospholipids which is in the inside of the cell membrane. So polar molecules cannot get through the non-polar side. Okay, so very well. Heads are polar, tails are non-polar. The usual structure is phospholipid bilayer, or basically what bilayer means, that we have two layers. Nevertheless, we mentioned an example when some living on or some living organisms can have also phospholipid monolayer. Do you remember what kind of living form it was we discussed last week?
Archaea has monorail.
Archaea, yes. Not all of them, but some archaeas can have a phospholipid monolayer. Then, and that is one of the factors why are they able to live in such extreme environments as hot springs, for example, or very acidic soils, or in very, very high or low temperatures environments. Okay, okay, sorry. And yes, so in this case, the correct answer is a C, phospholipid bilayer. Um, Regarding phospholipid quart, quat, quat. I don't know how would it be written, something with quattro or quart, I guess. I do not know about any example that some of membranes occur in four layers structure. So, and I think it doesn't exist, but four is definitely wrong. Nevertheless, some of you went for four. Hopefully, hopefully it's clear that we discussed also the picture and the structure of phospholipid bilayer. So hopefully it's now clear that C is correct option in this case. Can we move further? Yes, guys? Okay, let's go further. And Yuki, please, can you read?
Yes. The process when the cell engrafts bacteria is called a phagocytosis, B. Pinocytosis, C. Excytosis, D. Osmosis.
Thank you, Yuki. One more person. Okay, so here are the results. Let's discuss them. Yuki, what have you chosen and why? Yuki, do you hear me? You have to unmute yourself. Is not working, Yuki? Maybe Yuki, you can try to refresh, and then we will try whether it's working or not. Okay. Meanwhile, now please, what have you chosen and why?
I chose A, but I think B is correct too. But A is like a cell eating system,
Perfect.
and using shoe. I don't know how to pronounce it, but like using like hands and eating
Perfect.
is phagocytosis.
Yes. Exactly. So let me remind you the picture. Um, it's here phagocytosis. We are able to see pseudopodium. Pseudopodiums are exactly. Kind of imagining like hands or kind of limbs. Basically, those are um, um, the structures or created from or formatted from cell membrane. If we have a kind of material or bacteria and we want to phagocyte it, we create those pseudopodiums and we try to surround it close and get inside to the cell and then try to metabolize it or get rid of it if we are talking about bacteria. Only specific types of cells are able to phagocyte. We mentioned or macrophages or neutrophiles. Those cells are very important for our immune system. And on the other hand, pinocytosis is a kind of endocytosis as well. So, active transport way, which each cell is able to do. And what is happening basically in pinocytosis? That we have a surface, and it invaginates inside and gets inside to the cell like this. Uh, we call it also cell drinking. Phagocytosis we can call, as you said, exactly now cell eating. Cell drinking belongs to pinocytosis because the cells are able to invaginate only certain size of molecules, and pinocytosis or the pino part of a word is derived from Latin or Greek. I need to check, I'm not sure. However, it means drop of water. Okay, so it's just a drop. So very small particles, substances such as some hormones or nutrients can be drinked or pinocytosized inside to the cell. So only very small objects we can say. So both are active transport. Um, both belongs to endocytosis, and endocytosis is active transport. We also mentioned receptor-mediated endocytosis. Basically, what you have to know about it that is it already states in the title or in the name of it. So the endocytosis is mediated by receptor. It's a little bit more complicating kind of endocytosis, and it's not relevant for you now, basically. So everything what you have to know is just the name of it. And exocytosis or sodium potassium pump or other kind of protein pumps also belong to active transport. So, if we get back now, we discussed that pinocytosis cannot deal with bacteria because bacteria are in size much bigger. Also, we mentioned that phagocytosis is the process or way how a cell can deal with bacteria. Exocytosis is the way of active transport, however, it's the other way around. So, when cell itself produce something, for example, hormones or enzymes. And we used as an example uh, pancreatic cells. Pancreatic cells produce insulin, and if we have a pancreatic cell here and it produces insulin inside, it creates a vesicle. This vesicle docks on cell membrane and they fuse their membranes. So in order to open the space which was inside of the vesicle and release the content, really or relieve the content of a vesicle. Pancreatic cells can be an example, or other glands. Hypothalamus. It's a neurostructure which is very very important regarding the water balance, for example. So those can be just in examples where the exocytosis is happening. Um, yes, exocytosis. What do you think about osmosis? Is it an active way of transport or passive way of transport? What do you think? A is active, B is passive. One more person.
Sorry, I think I mistake.
Yeah, it's okay. Uh, Here are the results. I guess which one you wanted to click on? A active or B passive?
I think passive is correct, but I pushing A button.
It's okay. Never. It's okay. We will get to the right answer now. Okay, most of you went for B or passive, and most of you were correct then. So let me remind you what osmosis is. Osmosis is a specific passive transport, so it does not require energy. It does not require energy. And if we have here a semi-permeable membrane, yes, we have a semi-permeable membrane here, and on the right side there is a there are more or there is a higher concentrated solute, water and salt in higher concentration, and on the left side we have water and solutes in the lower concentration. Osmosis is a movement through semi-permeable membrane, movement of water molecules in order to get or maintain or reach, best word would be reach in this case, reach equilibrium. And equilibrium is the state when the concentration on one side of semi-permeable membrane is the same as concentration on the other side. So we do not need an energy for osmosis. What we need for osmosis is only semi-permeable membrane and the difference between the two sides of semi-permeable membrane, difference in concentration. Then the water will move from a lower concentrated side to the higher concentrated side in order, or thus it will dilute, so there will be more water, so it will dilute the solute and reached or get the balance of concentration or equilibrium on both sides. Understandable, guys? So far, hopefully now all of you understand why the question, where is the question? The question regarding osmosis was whether it is active or passive. The correct answer is that it's passive because we do not need energy to get an osmosis effect. Let me remind you one more picture. If you remember the activity from Friday, we were talking about osmotic effects in hypertonic, isotonic, and hypotonic environments. And if we have a hypertonic environment or solute, basically what does it mean? That the environment is more solute, the solute itself is more concentrated, so the water will go through the cell membrane from the cells through the cell membrane to the outside of a cell. So the cells will lose water and they will shrink. In isotonic, the exchange of water molecules inside and outside would be in balance. So this is the ideal situation. This is the ideal situation. And in the hypotonic environment or solute, the cells will um, fulfill themselves with the water, or water will pass through the cell membrane inside of a cell. They will be swelled, they will be bigger, and they can burst. Okay? So hypotonic is total opposite to hypertonic, and the direction of the water molecules is also in the opposite, absolute opposite way. Okay, and that brings us to the last activity. Let's have for this activity 2 minutes. Your task is to connect the points with the respective group of uh, types of transports. Let's have 2 minutes for that individually. Okay, guys, are we done? Yes, perfect. Okay, so what about the first point? Does not require energy. Where do you think it does belong to?
It's passive.
Passive, exactly. Misato, can you maybe link it?
Yes.
Perfect.
Mm, mm,
Or maybe on the mobile that feature is not available. I'm not sure. Uh, I can do it. If you have, maybe I don't know how does the Neuro looks like in the mobile app. However, up there is a white board in the computer. There is a pencil button, and when you click on the pencil button, you are able to draw on the screen. Okay, so next time you can try. Okay, um, but so far so good. Does not require energy. Belongs to passive. Great. What about osmosis?
Passive.
Passive. Exactly. We just discussed that. Perfect. Now, can you link it, please? Great. hmm Simple diffusion is next point. Where do you think simple diffusion belongs to?
Like passive.
hmm Simple diffusion is passive. Great. Can you link it, please, as well?
Okay.
Great. Okay, guys. Facilitated diffusion.
passive.
Great. Please, can you link it, Shoko?
Mm, di-
Great. Guys, before we get to the next point, what is the difference between simple diffusion and facilitated diffusion? If I will show you the pictures we saw on Thursday, what do you think? Simple diffusion, facilitated diffusion.
mm, facil-
Facility.
Whoever wants, go on, guys.
facilitate diffusion use transport the channel.
Okay. And
Pro-
and simple diffusion doesn't need any facilitator or helper, right?
yes,
Perfect.
yes.
Exactly. So more or less, they are almost the same things, but facilitated diffusion uses some kind of protein, carrier proteins or ion channels, basically via proteins via those the the diffusion is facilitated. Okay, so we they use a gradient. So in this case, though we can draw sodium here, it's in a higher concentration extracellularly, and in intracellularly it's in a lower concentration. But we know that highly charged ions cannot pass freely through semi-permeable membrane, so passage through semi-permeable membrane is not allowed for ions, so they need kind of facilitator, ion channel, to pass through it. So that's the difference between facilitated diffusion and simple diffusion. Very well. Okay. Um, what about endocytosis?
Active.
Perfect. Now, can you link it?
Okay.
hmm Require energy.
Active.
Very well. Can you link it, please, Sora, as well? And exocytosis.
Active.
Very well. Please link it, Choco, as well. Great. So, Very well handled, very well done so far. Guys, now that brings us to the core, and today's core is supposed to have two parts basically. First one will focus on semi-autonomous organelles, and the second one will focus on metabolic pathways. Nevertheless, it's 17:54 your time, right? Am I right? Okay. I would ask you whether you want a break or no. Hmm? Okay, so all of you want a break. Great. I suggest, guys, that we will have a break now, and then we will move to the core with the following activities. Okay, but it doesn't make sense to start the activities for core now and split them with the break. So let's have break now, and we will meet in 10 minutes, and then we will continue with the whole core. Okay, perfect. So see you in 10 minutes. Okay. Okay, guys, are you back? Okay, so guys, let me introduce you the core of today's session. Um, we will divide now into two smaller groups. You will work in group of 5 people. Preparation time will be 10 minutes. However, we can adjust that requiring your needs. Okay, so if you will need more or less time, it's up on you. Your task will be to focus on mitochondria for the one group, chloroplast for the other group. Focus on function and structure. Those 2 points are relevant for you. And that's basically it. I will provide you with the pictures inside the rooms, and the reason or why we are doing these activities again the same. I will just remind you why are we working in smaller groups, to create more space for a discussion, to collaborate with your colleagues and discuss your findings, because in a future practice or future studium you will many times work in a smaller groups, and you will study also in a smaller groups, and you will study things you don't know, didn't know before, and you will have to know how to deal with the others. And also in as a future doctors, you will deal with a plenty of other people, and you will also work a lot of times in smaller teams. So now we are just practicing it. We are getting into this environment, and this activity is, or the main message of this activity is to create a space in smaller groups for you in order to have uh, more space to discuss the things. You can voice out, so whatever you are searching for, whatever questions you want to ask your colleagues before we get to the main room, and then we will discuss together everything. Okay, both mitochondria and chloroplast. So that's it. So please, if you will be in these breakouts, try to speak a lot. Speak a lot, okay? Doesn't matter the language, whether you will use Japanese or English. Try to speak. Try to speak with your team members, okay? So to uplift a team spirit as well. Uh, okay, here is my presentation. Okay, let me now tell you who is going to do what. Okay, I will firstly need to check who is on the phone, because maybe some phone people. Okay, so okay, so I let's have it like this. Okay, so Misato, Mona, Satsuki, Yuiko, and Yuki, you will do mitochondrias. Okay, one more time. Yuki, Yuiko, Satsuki, Mona, Misato. You will stay here because most of you are using mobile phones, if I'm right, and at least what the neuro shows me. And you will do mitochondrias. Aoi, Nao, Shoko, Sora, and Yuta, you will do chloroplasts. Okay? Aoi, Nao, Shoko, Sora, Yuta, you will do chloroplasts. Do you have any questions before we start, guys? No? So let's start. Okay, guys, so remain here, and you can use this slide as an example. Okay, so please, I will turn off my camera and also microphone in order to keep you here private, and you can work together. Okay. let's start. Do you see a timer up there? Perfect, great. So you can start.
Come on. あ、これなんかコールインストラクターで多分ボタン押せばいけるんじゃないかな。
Guys, I'm here and I'm uploading the chloroplast. Or I can, or I will do it differently. I will just put you to the different room.
Thank you.
Guys, so here you are in the room for chloroplast. Okay, so this is the picture you can use. And yes, as I said in the main room, please try to collaborate with your colleagues. Okay, so this is the main message of the activity. Great.
Okay.
First, chloroplast diagram だよね。Diagram carry out photograph synthesis. あ、フォトシンテサイズだ、ごめん。光合成。
ああ、うん。
光合成。うん、フォトシンテサイズ。うん。で、in using 光エネルギーとかわざわざ言うのかな。ビームエナジー、ライトエナジー、
ライト、
using
ライト
ライトって光みたいな感じじゃん。なんかエナジーを強調して。
あ、光エネルギーってこと
光エナジー。うん。
ググったらライトエナジ
あ、ライトエナジー。
ライトエナジー。
using light energy and
ー。サンライトエナジーとかの意味かわかんないわ。
synthesize organic products.
これなんか、他なんか使うものとかいらんの？なんか、二酸化炭素やって。
どこまで詳しく言うかによる、ね。
そう。
ああ、確かし。どこまで言う？
協力隊だから緑色っていうの入れといた方がいいかな。当たり前すぎる。
あ、緑色、いいんじゃない？フォトは、color is green because フォト、違う、クロロプラストダイアグラムはず、その色素持ってるから。
うん。
なんだっけ？何の色素だっけ？
クロロフィルなのか。
クロロフィル。
からグリーンの色です。
で、このなんかグラナとか、チラコイド。
そこら辺、あれだよね、グラフにも書いてあるから。
あと、なんか、絶対いるのは多分、その、オン DNA を持ってること。
そうだね。
クロロプラストダイアグラム has own DNA and
形については触れなくていいんでしたっけ？
いや、ストラクチャーでしょ？
そうですね。
いや、言うよ。
これどう言うんですか？ めちゃくちゃな形してるけど。
なんだっけか、あ、違う、アキュミュレートじゃなく、
何て言い表せばいいのか。
なんか、楕円体って書いてあるんだけど。
パイルアップ
何それ。
積み重なる。
ああ、それはあれ、
グラナの、
グラナが
そうそうそうそう
グラナじゃなくて、
グラナ、
セラコイドが、
グラナ、
あ、チラコイドが積み重なって、
グラナってことだよね。
うん。
うんうん。
あ、そうだわ、ごめん。チラコイドだったわ。チラコイドが積み重チラコイズ、when チラコイズ piles up, when we call it グラナ。
グラナ
グラナ。
グラナ。
むずいむずい。
それかグラナはチラコイドのタワーによってできてるっていう言い方もできるくない？
ああ、いいね。グラナ、タワー。
フォーデッドストラクチャーみたいなかなと思ったけど。
ごめん、もう一回言ってもらっていい？フォ
ーデッドストラクチャー。
フォールデッドストラクチャー。
Group, would you like to have maybe more time?
Yes.
Okay, so maybe 5 minutes more?
5 minutes, please.
5 minutes would be good. 5. Okay.
Yes.
Okay. I will ask the other group and then I will add the time, okay? Guys,
イメージ。自分で。
guys, would you like to
いいと思うよ。
have
Two
more
membrane.
Guys, do you hear me?
Yes.
Would you like to have more time? The other group asked for more time, so if you would like to have also more time.
Yes.
Yes.
Okay, perfect. So 5 minutes. I will add 5 minutes. Perfect. Okay, so I'm adding 5 minutes and then we will meet in the main room, okay?
Okay.
Do you have any questions so far?
No.
Perfect. Okay, guys. So please collaborate, everyone. Be involved in the discussion. Great. Go on.
よし。
なんかミトコンドリア呼吸もできるみたいなこと働きで一応呼吸もあるんやけど、これも言っていいかな。 ATP
respiration produces
いるんやろ。
呼吸もできる。
呼吸ブリーズングなんかこれ自体が多分呼吸するっていうよりかはなんか呼吸するのを助けるみたいな感じの意味なんかな。 なんかミトコンドリアはもうユーカリオティックセル、真核生物にしかないっていうのも入れとくかな。
You can.
先生から習ったやつ一応
うんうん。
入れとくみたいな。
したら、それでいくと、エネルギーも必要ですみたいな。
エネルギー、あ、
require
ATP を産生するのにみたいな。
多分、
いるんやったっけな、これ。
なんか私の教科書には、that are very active and require a lot of energy って書いて。
require?
うん、require a lot of energy って書いてある。
じゃあエネルギー、can produce energy の後が、with なんやろ、with requiring その際いいな。with エナジーを作るためにエネルギーが必要だよみたいなことを言えばね。
何だろう。
ATP みたいな。もうそれを ATP にするか。どうしよう。
もうシンプルに、なんだっけ、エナジーを生産するのと、エナジーが必要な、なんだっけ、エナジーが必要な働き、エナジーが必要な作業ですみたいな。
ああ、じゃあもうミトコンドリアが働くにはエネルギー必要。エネルギーが
なんか、
いる。
ですみたいな感じだから多分。
うん。
マイトコンドリア can produce energy and require energy, require it みたいな。
おお、なるほど。and require。
もうどシンプルだけど。
いいと思う。じゃあ
じゃあ、葵くんよろしく。
了解です。
Okay, everyone. Hopefully, all of you had enough time for group work. Now, we should step into the second phase after the search phase or searching phase. Now let's discuss the organelles. So mitochondria group. Can we ask you to share with us your findings?
Mitochondria is only found in eukaryotic cells, and mitochondria has double membrane. In the inner membrane, fold the cristae and matrix. Cristae is folded structure. Mitochondria can produce energy and requiring energy. That's all.
Perfect. Anything else have you found, or anyone else would like to share something from your group, from mitochondria group? No, that's it. Okay,
So,
guys.
yeah.
Perfect. So let me maybe provide you with a little bit more information. So, but yes, you are absolutely right. As we already discussed, as you already know, mitochondria are only to be found in eukaryotic cells. And in the previous session or previous lessons, we discussed most of the organelles, with the exception of cell nucleus and so-called semi-autonomous organelles. Semi-autonomous organelles are mitochondria and chloroplasts. Why semi-autonomous? What does it mean, semi-autonomous, in the first place? Semi means half, and autonomous means independently. So it's halfly or partially independent organelle. What does it mean basically? Both chloroplasts and mitochondria are partially independent. Let's get a little check or insight or more details regarding mitochondria. Mitochondria, as you already know, and as you already said, they have their own DNA and their own RNA. Also, mitochondria have their own ribosomes, so they are able to do a proteosynthesis itself. They have DNA, they have RNA, and they have also ribosomes. So they are able to synthesize proteins. What we already shared today and also during last week, their ribosomes are more similar to prokaryotic ribosomes. That's related to the endosymbiotic theory we discussed, and we will mention it right now, I guess. Yes. So, what about their function? As Misato mentioned, they have they are let's say they are powerhouses of the cell, so they produce a lot of energy, but also they demand a lot of energy. So to have them or to make them be efficient and to work properly, they spend all or they need a lot of energy for themselves. Okay, so they have high energy demands. Where is the slide? Here is the slide. Yeah. Um, in some examples, almost 50% of the cell cell volume can be made of mitochondria. So here we have eukaryotic or normal eukaryotic cells, but in examples, for example, in examples like cardiac muscle cells or liver cells, mitochondria make up 50% of the cell content or on the cell uh, um, intracellular space. So almost half of their space inside of the cell is made of mitochondria. Liver cells or hepatocytes, we can call them, we call them hepatocytes, and cardiac muscle cells. Um, as as Misato's group already explained to us, we have two membranes, the outer one and the inner one. Now let me now demonstrate the endosymbiotic theory based on this knowledge or on these facts. Endosymbiotic theory, as you already very well know, says or claims that mitochondria were once independently living prokaryotic cells. Our ancestral eukaryotic cell was living also separately, individually. Then they made a trade business and they decided or agreed on that mitochondria will be engulfed and live inside of the eukaryotic cells. So basically, what happens, this engulfment is, we can imagine it like phagocytosis, what we already discussed. And as you see, I intentionally draw this eukaryotic cell in the red color. And as you see here, it was engulfed and even this was detached. So, okay, so what we had now was this is an outcome, let's say. I will draw that. And here we have mitochondria. And this red line represents the outer membrane of mitochondria, and the green one here represents the inner membrane. Here we are able to see it also in the red color. This red line, as you see, here it's a brownish layer, yeah, brownish, but the red line here should represent the outer membrane. And outer membrane is very smooth as it's permeable, so it's permeable to polar molecules like water, or but it can also have like specific channels called porins. We call it porins. It's specific channels. The outer membrane is, however, let's get back to the outer membrane. The outer membrane originated from the host cell's plasma membrane. Here it is obvious or apparent. Here we have an ancestral eukaryotic cell, and this was the plasma membrane of itself. When it was engulfed, okay, it was phagocytized basically, yeah, like this. We see that now the mitochondria are surrounded with the plasma membrane of the ancestral eukaryotic cell, and this represents current outer layer of mitochondria. Okay, we are able to see it even here. Plasma membrane, the outer plasma membrane of mitochondria are more similar to eukaryotic plasma membranes, and inner plasma membrane are more similar to prokaryotic plasma membranes. They already mentioned that, okay? So this is an outcome. The double layer of mitochondria is an outcome or result of endosymbiotic event or theory, let's say, okay? Let's now talk a little bit the intermembrane space. Here we have intermembrane space. What we have to just know, it serves as an space for transport the things from outer membrane to the inner membrane and the matrix. Okay. When we are talking about inner mitochondrial membrane, here the yellowish lines, and we are able to see a cristae here. We see how differently structured it is. The outer one is just a regular membrane. The inner one is very folded, so it's like uh, very curved. It's very folded, and it makes or it creates cristae. Cristae are structures or folds in order to maximize the surface. What does it mean to maximize the surface? Do you understand that, or should we discuss, or should I explain that? To maximize, I will use this picture. Here we have a surface. Okay, here we have a surface on a cell, basically. Okay, so on every spot on the surface, you are able to maybe somehow use it. However, the inside is not used. It's empty space. It's empty space. So a lot of space is not used. But if you do the cristae, so basically your cell will look like this, and you will do these folds and curves. You have additional space where some metabolic processes can happen. Okay, not only on its surface, but also on these folds. So your final area where the metabolic processes can happen is larger, basically. Let me use or show you analogy. I will share my screen. In other analogy of the folding or using the space much more efficiently is our digestive system or our intestines. So, for example, you see here how the intestines are curved and folded, you know, uh, and folded. So when you would have only a linear tube or a linear intestine, it would be like 6 meters long. None of us have 6 meters in the height, However, we have intestines which are very efficiently folded and placed just in our under our stomach, and they are very efficiently folded in order to maximize the surface or area where the food can be digested, but they are very efficiently folded, so they are distributed only on a small area. So that's another analogy of cristae. In cristae, when we are talking about mitochondria, let me get back. It's very similar to our intestines. Okay, mitochondria try to use the space inside of the inner cell membrane in order to maximize the space where the metabolic processes or pathways can happen. So that's the reasons of cristae, why we have them. Yes. So that was regarding inner mitochondrial membrane, and that brings us to the matrix. Um, uh, one more very important, one more thing to mention regarding inner mitochondrial membrane is that respiratory chain takes place, or oxidative phosphorylation takes place on inner mitochondrial membrane. In the matrix, on the other hand, Krebs cycle is happening, or yeah, um, is going on in the matrix. And in the matrix, we can find um, ribosomes, already mentioned ribosomes, or DNA and RNA of mitochondria. Also, enzymes, or yeah, of course, enzymes of Krebs cycle. Um, so Krebs cycle is happening there, and water ions and some mitochondrial compart substances or molecules. But the ribosomes, DNA, RNA, and Krebs cycle are the most important things for you to maybe remember, for you to remember regarding mitochondrial matrix. Yes. So what else to add? I think that's basically everything necessary for you. Yes, I guess that's basically it. Okay, guys, do you have any questions to mitochondrias? No. Okay, so maybe one more thing I will mention. On this picture on the left side, let me just erase my attempt to draw. On this picture, we are able to see mitochondria in a real picture from electron microscopic perspective. So, as you see, here we have an outer membrane of mitochondria, and here those lines inside are crystals, what we were discussing just right now. Okay, crystals are those things in mitochondria. Perfect. Okay, so that's it. Thank you very much, group. So thank you very much, Misato, Mona, Satsuki, Yuiko, and Yuki for your research. And now let's do chloroplast part.
Can I start to talk about chloroplast? Can you hear me?
Okay, guys, so can you share with us something regarding chloroplasts?
Can you hear me? Some
Yes, yes.
Oh, okay, nice.
Sorry, I didn't hear you before. Maybe if you were asking.
Yeah, okay. Yeah. Chloroplast is only found in plant cell, and it does photosynthesize by using sunlight energy and CO2. And it is green because it has chlorophyll.
Mhm.
It has own DNA and has two membranes called outer membrane and inner membrane. And inside it, there are many grana, which is made up of stacked chocolates. That's all.
Okay, thank you, Aoi. Anything from Aoi's group to add something, maybe, guys? No? Okay, thank you very much. Yes, so I think, yes, you mentioned the essential information. So let me maybe provide you with a little bit more things. Where to start? Okay, so chloroplasts are also semi-autonomous organelles, as mitochondria are. So, however, they are typical only for plant cells, as Aoi said. Um, Chloroplasts are organelles belonging to the group of plastids, and the plastids or chloroplasts are responsible for photosynthesis and the Kelvin cycle in all plants. Kelvin cycle is maybe a new term for you. You don't have to bother much with that. Nevertheless, just note down that Kelvin cycle is related to photosynthesis in plants. Okay, so Kelvin cycle, something like this can occur in admission questions. Okay, that's everything you have to know so far. As you very nicely says, it has two membranes. Outer membrane, as we are able to see on picture, and inner membrane. So this is a similarity to mitochondria. Two membranes, and the reason why chloroplasts have two membranes is the same as in mitochondria, because endosymbiotic theory applies also for chloroplasts. So the story behind is basically the same. Chloroplasts were once independently living and they were engulfed by eukaryotic cells, or in this case, specifically plant cells. Yes. Regarding the structure, what is different? The inner environment in mitochondria we call matrix. However, in chloroplasts, we call it stroma. Okay. Then we are able to see some other structure we haven't heard before, and it's still there are thylakoids. And thylakoids are basic functional unit. It looks like a coin, basically, okay? Like a coin. Thylakoids are basic functional unit where photosynthesis takes place. Okay, so here in this green coin, photosynthesis takes place. And if there are more thylakoids, stack of them, so there are more coins above themselves on each other, they create some kind of stack. We call the structure granum. Okay, granum. In addition to photosynthesis, there are more characteristics to mention. First of all, I would mention the pigment containing structures. As you said, as our group said, chloroplasts contain pigments called chlorophyll, and chlorophyll gives plants the typical green appearance or color. And chlorophyll is responsible for capturing light, okay, during photosynthesis. Other pigments can also be in the chloroplast, such as carotenoids, for example. However, they contribute to the colors of the fruits, for example. Yeah, but if we are now having just this picture and we are talking about plant cells, let's keep it simple. Chlorophyll is responsible for photosynthesis, and it gives plants the typical green appearance. Yes, now uh, the production of ATP. Photosynthesis is using thylakoids, or the thylakoids membranes have like many components involved in the photosynthesis. However, what is important to mention, or what is important for us, is the outcome. And the outcome are that we have energy from it. We have energy in form of ATP. ATP, we already mentioned that ATP, or adenosine triphosphate, is a molecule very, very important for living forms. And one of the best ways how the living organisms can store, produce, and use as well, or utilize energy. ATP is a adenosine triphosphate molecule, and I will upload the video regarding that after the session, so which you can watch as a homework. So that will be only one thing for your homework for tomorrow, okay? So ATP video. Why is it? You will understand me a little bit better what, or yeah, in what way or how important the ATP is. Nevertheless, we will just tell that it's very important. And here in these thylakoids, the ATP is produced due to or thankfully via photosynthesis. So it's very, very important, those structures and chloroplasts. Yes, what else? Carbon fixation. Um, in this process or carbon fixation or the Kelvin cycle is happening in stroma, and the result of it is all are also ATP, are ATPs. So yes, you can know that. I don't think uh, it's so essential for you so far, but for those who are interested, you can have a note that Kelvin cycle is happening in stroma, and Kelvin cycle also produce ATPs. What I think can be interesting for you is plastic differentiation. Plastic differentiations is that chloroplasts are plastics, as we already discussed, and they can have like distinct structures and functions. And however, they can change themselves in another type of plastids. So chloroplasts can change themselves into chromoplasts or amyloplasts. Chromoplasts are involved in pigment synthesis, for example, or amyloplasts. And they are totally different organelles. However, they can change themselves based on the conditions or based on their needs. For example, amyloplasts are storing starch. Room chat. Please check a room chat. There is in Japanese written starch. Do you know this word or still no? Starch, or in Japanese here is written, I don't remember how it's what is the name. Nevertheless, guys, starch is nothing else like a lot of we have a glucose molecule, and the glucose molecule is linked or bonded with another glucose molecule. So if we have a chain or a lot of glucose, yes, next to each other, like a lot of glucose bonded together, we have like a polymer of glucose or carbohydrates, we say, and we call that starch. And it's much more usual in nature to find glucose in this form. Yes, what I mean, uh, starch is a component, for example, in potatoes, rice, oat. uh, I will show you the picture I also used in previous session. So here, let me show you. Desktop share. Here we are able to see sources of starch. Okay, so guys, now do you know what the starch is basically? It's a polymer of glucose. Yes, So, and here we have typical resources like beans, brown rice, muesli cereals, oats, potatoes. Yes, they are very rich for starch. How we get there? So remind me. Amyloplasts can store starch. So if there are some conditions in the plant life when we need to store a lot of starch, chloroplasts can change themselves into amyloplasts, okay, or chromoplasts. So chloroplasts are very adaptive organelles, and that gives plants the very um, evolutionary useful or beneficial skill that they can survive even struggling times. So they can adapt much better to the current conditions than, for example, animals or or humans. So this was just the interesting information, I guess, hopefully. And yes, so that was regarding this. Regarding endosymbiosis, we already discussed that they were also engulfed. So guys, as a summary, I would use this statement I have written here. I got written here. So chloroplasts play a crucial role in the energy production and metabolic processes of plants, serving as the site of photosynthesis and contributing to the growth and development of green organisms. So this is like one longer sentence to summarize chloroplasts. They play a crucial role in the energy production and metabolic processes of plants, and they serve as a site of photosynthesis, and they contribute to the growth and development of green organisms. Not only plants, also some animals which have chlorophylls, but this is not important now. Let's talk about like plants. Let's imagine like plants in easy way. Yes. Okay, guys. That's it. Do you have any questions? No. Okay, so we discussed chloroplasts and mitochondria. Tomorrow we will get to the second part and we will have a look on metabolic pathways. Guys, you don't have to bother a lot with this topic. This will be just overview because metabolic pathways are the topics for you in the second year of medicine when you will be dealing with biochemistry. Nevertheless, it will be very helpful for you to understand how the cells are creating or producing, metabolizing the energy, and we will discuss this tomorrow. Uh, do not bother if you will want to do your own research in advance, you are able to, but do not be stressed out when it will be very complicating for you because there are plenty of um, very hard chemistry patterns and steps, which are the topics for you in biochemistry. Okay, so it's we are still trying to understand and comprehend the basics. So do not be bothered with that, and I suggest that you will keep that for tomorrow's session to have an only overview. Your only homework will be the video, to watch the video about ATP, and I will upload it to the Google Classroom, okay? So that's it. And yes, then we will get there tomorrow. It's 12, it's my time. So it's 7 p.m. your time, right? Perfect. Okay, guys, so can you maybe give me a very quick feedback about the session? How did you like the structure? What are your feelings? If you would like to keep the structure like it is, that we are starting with some recap of the knowledge we already gained, then we have some active activities where you are collaborating with your colleagues and so on. So what are your feelings basically? If you can express them via emoticons, it would be great. two more people if you can. Okay, perfect. Mhm. Okay, great guys. I'm very glad that you are satisfied with that. Hopefully it is also beneficial for you. That's everything from my part. Thank you very much that you were again very active and that you were collaborative. So yes, that's everything from my side and looking forward to see you tomorrow. Have a nice rest of the day, guys. Bye.